in the soil around the root exudates would go out. They would send sugars that they made through photosynthesis to feed the microbes. And then essentially it's a barter system. The microbes make certain nutrients that the plant needs. The plant makes certain nutrients that the microbes need, and they trade them back and forth, just like a medieval village, right? There's a baker and a cobble person, and they work together. When plants are fed totally liquid nutrition using chemical inputs like fertilizers, they don't have to work so hard. They get a little bit lazy. Those of you who can see the slides can just see a quick little picture of a couple plants, a control plant, and one's fed phosphate, nitrate, ammonium, and potassium. And you can see particularly when they're fed chemical nitrate, they don't need to grow root hairs because they don't need to mine those microbes to give them nitrogen from the soil. They can just sort of slurp it up, right? It's like giving kids sugar. It's easier to eat than broccoli. So supplying them by nutrient chemicals alone really does sort of break that cycle between plants and microbes. And it's very hard to get back. It's really the issue. Healthy soil is dependent upon the microbial community. And there's, of course, on the internet, tons of stuff you can watch and read. I'm sure no one has time for that, but plants, microbes mine and produce minerals and nutrients that plants need. Microbes are the ones that decompose the litter of plants, roots and other things. And though that litter becomes soil carbon, which then becomes that catchword, right? Sequestered carbon in the soil, so that it also stays in the soil, but that soil organic carbon can be food for microbes when times are lean. So it sort of builds food security in the soil. And microbes, more importantly for growers, I think everyone cares the most about soil structure. So microbes, particularly fungi, secrete a substance that has a sort of a boring name, which I forgot right now, that's sticky. The microbes stick to them little particles of sand and clay. They create little pockets in there to live. And in those pockets is where little droplets of water and air live, right? And as the microbes die, that sticky substance stays. So as you year over year have microbes cycling through your soil, you have more and more of this soil structure, which increases water holding capacity, resistance to drought, and is much more resistant to erosion. So this year in particular that was so dry, areas that had very low microbial counts were not as resilient as those with very high ones. Plants and microbes also sort of communicate with each other. We're not exactly sure how, but they do communicate things like threats and pests, and trade sort of immune chemicals that they need. And they also sequester carbon, which I mentioned. The important thing to understand about a microbial population, and one thing people ask us a lot about, is to identify bad microbes, as if they're sort of, two camps, but that's not quite true. Microbial population is quite complex. Microbes have very, very little DNA, and they usually can only do one or two things. So they're dependent upon each other to barter and trade. So as microbial populations grow, they get more diverse. This is just a slide. The first day, you can see on the left, we put bacteria on it and nothing grows. Over time, you'll see all different kinds of communities start growing. And that's because, say, bacteria A starts to grow and it makes a substance that bacteria B needs. So then you have bacteria B. Bacteria B will make a substance that bacteria C needs. So when we think about soil microbes, we sort of equate size and diversity together. And then we equate diversity with resilience, because I'm not going to get into it, but it's kind of a cool concept of quorum sensing. It's when sort of, maybe not bad microbes, but microbes that can be pathogenic. If they get to be big enough, they can sort of attack. But if you have a microbial population that's diverse and big, it's less likely that those pathogenic microbes can get enough of them together to cause problems. This is, of course, not to say there aren't bad bacteria and diseases and all kinds of problems. we're not trying to be too naive about it. But that's sort of in the ideal world, the microbes would, for the most part, regulate themselves a lot. This slide is just more about building soil structure. Again, you need microbes growing all the time in your soil. One of the big pushes of regenerative ag is no bare soil. And that's because as long as something is growing or there's roots and detritus in the soil, microbes will have something to eat. They'll continue to reproduce and grow and die and build soil structure so that all year long, for the most part, I've been in Minnesota, it's probably not a lot of microbial activity in February, things are growing. So that's it. This is another picture about the soil aggregate cycle. Okay, again, how microbes build soil structure over, and the you know, as microbes die, some of that structure falls apart, but if they keep growing, they'll keep going through them. It's this, for those of you on the phone, it sort of looks like a nightmare biology textbook for when you were in school. One other thing that's very important and that microbes allow is microbial food security. So carbon stores in the soil. So as you leave roots and dead plants on the soil, microbes eat them. The microbes are about 60% carbon. When they die, that carbon is in the soil. And then their bodies either stay there or get consumed by new microbes. So that when there's not a lot going on or when there's stress and drought, if you have really good, long-lasting microbial communities, you'll have more carbon stores in the soil for resilience. In terms of soil carbon, there's fresh and stored. They're very complex. Everyone has tons of different names for them, but it's sort of the difference between like readily available food and food you have to cook. You know, soluble, fresh, stable organic matter is things that plants can consume quite readily, and stored or stable is things that are harder for microbes to eat, but they're still there. Priming is one thing that we sort of talk about a little bit. If you have soil that's very, very poor and you throw down, say, a microbial input, there are some terrific microbial inputs out there, don't get me wrong. But if you have soil that cannot sustain microbial life, you can't just throw down, you know, a bunch of molasses, some compost tea, and hope that they'll live. They'll eat what you gave them and then die, you know, which is why when we talk to people in the field, groups that use things like Growers Mineral or other biostimulant that sort of improve the food supplies and the nutrients for plants and microbes, you get a much better sustained boost to both soil health and yield. Building a balanced microbial community is really, really difficult. I think you guys probably know it certainly more than I do, that farming is particularly difficult now because of both the economic pressures, the sort of supply chain nightmare that you're dealing with, and then the regulatory and the consumer demands on you. Ideally, sort of easily, we say the goal is to create an optimal conditions for microbes and plants to thrive in the soil, but that's easier said than done. It depends on what's happened to your soil, where you are, what the soil is made of, and what you're trying to grow, okay? But we do know that feeding microbes and getting soil the right level of nutrients and calcium can start that cycle moving again, okay? And what you want to do is start the microbial plant symbiosis moving so that all those good things can happen to the soil. So this slide is got a lot of words on it. Those of you on the phone, it's fine. We just talk about solutions. So right now, as we're trying to grow plants on soil, there's a lot of things that people do. We put down chemical fertilizers, right? There's certainly a place for those, and probably always will be. They are incredibly effective for improving yield. But as we do know, over the last 2 or 3 years, supply chain, global things, and price are making them very difficult. And we also know that if we use the microbes and make the soil healthier, we can use less of them. So it could be better for your pocketbook, it'd be better for the earth, be better for the people and everything. Microbial inputs, I mentioned those. There are some good microbial products out there. It's sometimes hard to know what to use where, okay? But a lot of people use mycorrhizal fungi in particular to help build soil structure. Research is ongoing, but it's sort of hard to know what will work where. And if you're using those, you should be doing quite a lot of research to make sure you're not just throwing down these poor microbes, spending your money on them, and then letting them die. Cover crops, so that's, I don't know if that's something that's very common in the group, but that's sort of the mantra of eco-ag, regen ag, is no bare soil, right? At least keep some litter on there so that microbes have something to eat. The soil doesn't blow away, and you hold some moisture into the soil even when things are dry. And then biostimulants, grower mineral solution can be considered one of those. They work because they provide better conditions for microbes to grow and then for plants to thrive on the soil. Okay, so those are more effective, we've seen, when people are trying to rebuild soil microbial life and structure, because they sort of improve the general conditions of things. Optimal conditions, people ask that a lot. I have no idea. It depends on a lot of things. You probably know that better than I. Soil composition, pH, compaction, how much soil organic carbon you have in your soil, temperature, seasons, crop history, water availability, right? Some of these things change year to year. So it really is an art rather than a science. So I know I went over those so quick. So strange not having anyone nod or write back to me. And someone is
actually, Laura, I have a question for you.
Sure.
Are there any, besides cover crops, are there any other kind of best practices, general practices that would help regardless of where somebody is in the country, what kind of soil they have, the history of the farm, and so forth?
Yeah, there's a lot of much better people to ask, but I will tell you what I've heard, and I hope I can remember all 6 of them. So sort of the 6 general principles that people have for regenerative ag, and some of them are easier to do. They're all hard to do, I'm not gonna lie, but some are, to me, seem somewhat impossible. The first is sort of no bare soil, right? As best you can. In some areas, that's very difficult just because of timing, cost, and machinery. But no bare soil, so keep something growing or on the soil at times. Limited disturbance, right? So for a while, there was the battle cry of no till, right? But as all pendulums, that sort of swung back. And so now people use sort of terms like selective till or minimal disturbance. But you want to do very thoughtful disturbance of the soil. When soil is disturbed over and over again, carbon gets released, which is the stores for the microbes, right? And the fungal connections and the fungal life in the soil, which really creates a lot of structure, gets disrupted, okay? So there are times when you're going to have to disturb the soil, but sort of the constant massive tilling really does sort of ruin all the homes of those microbes. The third is, oh gosh, I don't know, 2. Those are the 2 big ones. Livestock integration is another one that people try to do. When I hear people talk about it in real life, it looks like the hardest thing I've ever seen, because particularly cattle have to go through a field very, very quickly. So unless you want to spend your life on a horse, herding cattle around, I'm not sure how possible that is. Limited use of herbicides and pesticides is another one. Again, it's not saying they shouldn't be used, but it should be quite judicious. Same with chemical fertilizers, right? Only when necessary. And I think I just went to a really terrific presentation that went through it, but I don't have it on me. But those are sort of the easy ones of regenerative ag, but it sort of depends. People are also into intercropping or, reducing the amount of monocropping there is and increasing diversity in any way that's possible on farmed land. So diversity of crops, either in a year or throughout the year, or even some people do intercropping in rows, which again, good for them, but that seems very difficult.
Yeah, that would be very labor intensive.
Yeah, I'm not sure I'd want to be doing that. But you know, some people do that, and you know, it's pretty hard. When you talk to a lot of these guys who've converted, you know, a lot of the companies will say you can, you know, in a year convert, but really most people who've done it say if you can do it in 3, it's pretty impressive.
Mhm.
And 5 is a little bit more realistic. But even people taking small steps makes a big deal. I mean, I think 3 to 5% of the US uses cover crops. And so even increasing that a bit would make a big difference.
Yeah.
Yeah.
Do you know,
So, mhm.
um, do you know anything about the effect that, like, one of the things that growers talks a lot about is high calcium limestone application on the soil. Its benefits are well documented both in our research and outside of it. But one of the biggest, well, my general question to you is, do you have any knowledge on the effect of calcium itself on soil microbes, or the effect it has on soil benefiting them? Because like for
Mhm.
context, one of the big things that we've seen is that it allows better air and water infiltration into the soil. It flocculates the clay.
Yeah, yeah. So, you know, I'm going to say no. I wish I had a great answer. But, you know, it's funny because I went to this talk and they said, you know, everyone bashes clay, but clay's terrific, you know. But everyone wants just the right amount of clay, right? And most people either have too much or too little. So if you have too much, then something like the calcium flocculating would be very helpful. So I can't speak particularly, and that's where it comes down to like people having to understand what's happened to their soil, what they need in the soil, right? And that's where the testing comes in, both chemical and biological testing. And I think when Jim was still with us, he mentioned, you know, testing soil is hard because soil it's not a vat that we manufactured, right? That you can just mix up and it's the same everywhere. Even in a small field, there'll be differences throughout the soil. So we try to do things like take composite samples and do all kinds of stuff. But really the answer is sort of systemic and frequent and often testing, you know, gives you enough data points that you can get a real story from the number instead of once or twice. So, and, you know, we're in the soil testing world. So we always have a hard time with people being like, I took one test and, you know, now I need to know what to do. And we say, well, you know, that's just the first start of learning about your soil, is seeing how it reacts to things over time. So if you hear my speech or you're already into it and you know, want to increase your soil microbes, you have to know that throughout the year, they go up and down naturally. So when the plant starts to send out exudates and bud, you'll have a lot of microbes. When the plant goes to flower, the microbial population will naturally go down, right? Because the plant is feeding its babies, doesn't want to feed those moocher microbes anymore. And then, of course, in the winter, microbes will go dormant. Microbes reproduce, some of them as much as every 15 minutes. Okay, so it's not like it's going to take forever to bring them back, but you do need to have the soil conditions. And microbes are everywhere. So literally we say, don't worry, they will fall from the sky onto the land. So don't worry too much about it. Again, there are situations when certain microbes need to be added to soil, and that's something that someone's going to have to figure out. But it is much better when you make soil conditions, you know, if you improve water retention, you'll improve microbes and plant life.
Yeah.
Hey, Laura, Matt Gooding.
Yeah. Hey, Matt, how are you?
Behaving. Laura, you know, and I'm sure you're aware that there are so many multiple products out here that supposedly a farmer can add biological products. Call them bug in a jug. They've been around 40 years. I remember my dad messing with some, growers messed with some through the research department 40 years ago, and they just seem to repackage it and remarket it and kick it back out there. Now, why sometimes there seems to be responses, consistency, is nothing. There's nothing. What are you
Yeah.
seeing, or what kind of feedback you getting? Can you
Yeah,
elaborate?
so I'll tell you, we get feedback from people who are using our test kit to assess the impact of inputs on microbes, right? So we don't have any products that we sell besides the test kit. So, but over the past 3 years, the bug in the jug thing was everyone's, dream a couple years ago, right? And it was, you get one and it worked amazing where it was developed in Tennessee. And then it couldn't roll out anywhere else, right? For a number of reasons, right? Maybe that microbe was missing in that soil, or maybe that might and can't live somewhere else. So for the most part, most people who get sustained and long-term improvements in yield and soil health, right? Because you guys aren't mining microbes, right? It's not like you're just excited that you have more fungi in the soil, right? You want to turn it into fertility and yield, are biostimulants. And I use biostimulants, but there's lots of different words. But products like Growers Mineral that do things to the soil to make it a better home for both roots and microbes are much more sustainable. And we do know that microbes and plants sort of all over the world eat the same thing, right? And we're not quite sure, but there are billions of microbes, right? But early evidence says that about 70% of all microbes around the world are the same, right? Which means that, there's some local variability, but more commonality than that. And again, they're in the air, they're all over us, they're all over, even seeds when they germinate are covered in microbes. So it's sort of like the health craze that went through, you took bacteria in your gut, what were they? Probiotics. And now people are finding that really, if you want to improve gut health, you eat prebiotics, right? Things that feed the microbes that you already have. And we're finding that with soil as well. But, people still sell microbes. Big companies are out there doing all kinds of genetic testing, trying to find the world's best super microbe to, I don't know, resist Roundup or something. So people are working on it.
So Laura, what do you need? You're interacting with the farming community and this the microbiometers out here, and you're finding all this information. What are you looking for to kind of improve from your point of view this device being out in here? What do you look from farmers? What kind of data do you like? What do you like to see?
So one thing, and our users are amazing. They call us if they're happy, sad, bored, all kinds of things. It's great. There's only 4 of us and we all work at home, so we're happy to talk. So a lot of what people do is they call and they say, this weird thing happened, right? Like I saw a big jump in microbes, but I didn't see a jump in yield. And we sort of talk about why that might be. Or we talk to people who say, particularly like we're a US-based company, but we sell mostly abroad. So people call with sort of strange soils around the world. Our real dream is that growers would, systematically test their soil and figure out sort of the natural cycle of their soil and what works to improve it, right? What dosage and what products and what crop practices are sort of the most realistic, right? Of course, there's an ideal. We're all going to mob farm and holistically weed, but that's just not possible, right? We can't do that and stay alive. People need to sleep at times. But you can say, okay, these are the big hitters that'll help me both, reduce costs, improve yield, and bend sort of to the both regulatory and the consumer pressure to decrease use of chemical herbicides and pesticides. So we love hearing from people that use it. We find glitches and problems in our test too, which is always sort of sad when we find that out, but it's wonderful because people tell us and they're very forgiving and then we fix it. So it's always good to hear what things don't work. For example, the iPhone 14, terrific phone, but we're having problems with it. So we're doing a patch right now. The iPhone 14 is too good a camera. It keeps wanting to make our test card looks prettier, I think, or put a filter on it. So we're working on that. Any other questions? Is there anyone? I don't know. You do *6 if you want to talk. Feel like
Sometimes our
Ferris-
listeners like to play hard to get.
I know, I feel like Ferris Bueller. Anyone? Anyone? out there?
Yeah, I
Bueller.
got a question for you. Can you hear me?
Yeah, I can hear you.
I would just what kind of effects fungicide has on microbes.
Well, fungicide kills.
Everybody's fungicide crazy now.
Yeah, fungicide kills fungi, right? So it's not great. Anything that is designed to kill living things will kill living things, right? We're big, so fungicide that gets on us won't kill us. But if you dipped one of us in fungicide for too long, we might die, right? Wouldn't do that, but yeah. So I don't know offhand, but fungicides and herbicides and any sort of treatment that's designed to kill things will have a negative impact on the microbial population. Now, sometimes you have to use them because you have, disease. But if your soil is resilient, hopefully you'll kill the sort of, right, the bad actors in the population, let the population re-homogenize and go healthy again. But what people try to do is use limited use or do it, only where it needs to get done and not do it preventatively, use it as a real precise tool, not just sort of a, every year you just throw the same things down no matter what happens.
I don't know if it's just because of the availability of them, but here the last few years seems like it's getting to be almost a normal practice. I didn't know if that was maybe something that was working against the soil, or
Yeah, I mean,
why would
I don't have any real
the fact
data
that
on it, but
yeah.
pardon? Sorry.
Oh, just the fact that it seems like they basically think they need it anymore. I mean, you hear these guys are all wanting to use it.
Yeah, you should use things that you definitely know you need when you know you need them. But again, we sort of beat the drum that if you do things to improve your soil resiliency, you'll be less likely to get disease, right? Not that you won't ever, but you won't have to use so much of it so often, which is better, right, for everybody. You don't have to worry about buying it or getting it now. Yeah.
Laura, one of the experiments that we did here in the farm was basically we did a check, and then we had an area where we had used a dry fertilizer and growers, and it was done in October. It was done on a hay field. We took conventional soil tests, we did forage tests, and we did biometer tests on the soil. And ultimately, we did them 23 days apart too. So because the soil was warm for this time of year in Ohio, and we were able to do, I think it was 21 or 23 days apart. And the reality of it is where growers was used, we've seen a increase in the difference in the numbers in the tests. Now, I look at the microbiometer test as a comparative test. So it's important how you set up protocols and the test protocols to do it. So you do get, a comparison. It isn't so much the number as how that number goes up and back and forth. And that's how I think of this. And if I'm wrong, we have to have a conversation. But as so often happens, it's the things you don't think about that you do that that kind of surprise and lead into something else. And the soil test that I had taken, the soil I had stuck in a jar and stuck in the cupboard in the lab through the winter. And a few months later, I took those tests out again, the check and where the dry fertilizer was and where our growers was, sprayed on through the leaf and watered in those soil areas. And so basically, this is the classic soil that, I mean, in a lab, it's sitting in a, it's not subjected to good aeration, weatherization, 60 degrees or below, probably in that cupboard, in that location through the winter, dead, you know, not no light, no plant food or anything. I just took them out and I rerun them through your biometer and was just shocked at how, we were still so much higher over the check where we had used this growers. Okay.
Yeah, yeah.
And so, and, you know, please speak and elaborate. My mind goes into when I seen that, the longevity and building of that carbon, you know, when I rehydrated that soil, basically, because it was dry soil, are we measuring the dead carbon part of the bacteria from that experiment, or are we still measuring the live portion?
Yeah, so that's a great question. I'm going to address your first comments first for everyone. Our test, the microbiometer, is designed to be used in real time on living soil. So you don't crush it up, dry it, you know, keep it on a shelf for 6 years and measure it and think that that's the conditions in the soil when you took it out, right? So it's like if you catch a fish, right? You weigh it right then and there. You don't dry it out and then weigh it and so say you caught a 12-ounce fish, right? So you take the soil, fresh field moist soil, you measure it within a couple days. Now your experience of taking the soil that you had treated to improve its microbial life had much more spores in it, right? It was healthier when you started to dry it out, right? And it had more microbial life. So our test measures living and dormant microbes, not dead microbes, because they sort of fall apart and degrade. But when you treat soil and it's healthier in general with more microbial life in it, when it gets stressed and the microbes die because of drought, there'll still be the dormant and spores in there so that when you rehydrate it or the spring comes, it will bounce back faster, which is why you got those better results, right? Does that explain it?
Yep. Nope. Of interest. Proximity too. One of the things we're doing is we're finding that with our fertilizer placement at planting is running, I would say we're just going up another level. In other words, we would always put this in around the seed. We always felt that we were not all about fertilizing a plant. We've always known and felt, and until your biometer came along, we were able to prove that we do improve soil microbiological activity, which is awesome. I mean, you know, there's this company's been around for generations. I'm second generation. A lot of the people listening are second and even third generation growers, users, and their dads would always say, "Yeah, this stuff seems like we get, you know, it's got to be helping the soil microbes." We could never prove it. And thank you for, you know, you guys having this, because it has helped us realize what we always wondered and thought. We could never put it in paper. So it's kind of neat. So what we've done is we've said, okay, so if we're improving this microbiology here and we can see it, you know, what happens if we put at planting our product in multiple places? And we seem to be going up a whole nother level. So in devising experiments with the microbiometer to do that in the future, are we how am I put this? I'm trying to think how to do something real time in the field and devise an experiment in such a way that that we can show that placing it in those number of spots that we're getting this actual yield. I mean, customers are modifying their planters. I mean, we're on it.
So the placement of the product vis-a-vis the seed or the roots, right? Because if
Right.
what you're doing is improving microbial viability, the closer to the roots you are, the better it is, right?
Well, we're
sort of.
spreading it out though. We're finding out to take the
Oh,
same
really?
volume. We're
Oh,
taking the
neat.
same volume and now we're putting it in 3 different spots, and now we're going to a whole nother level. We're actually adding water, and so one of the things I'm going to be doing this winter is figuring out if the type of water means something, whether it be rainwater, reverse osmosis, well waters, how that reacts with microbes. We're going to have some interesting things, I think, come up.
Yeah,
So
yeah,
I'm just trying to glean, how do I want to say it? I'm trying to pick your brain.
yeah, I mean,
for
we
you
would
to,
have
for you,
we should have a deeper conversation about each one, because
maybe.
it is hard to sort of devise a test. We've had a bunch of academics call us, and I know this is probably not a pro-academic group, so I can say this. We had an academic slam our product and said it didn't work, and we called and said, what happened, because we had talked to them, and they said, well, we did the test. And I said, well, they were comparing 2 tests together. They tested the soil a year and a half apart. The soil biology. And I said, I don't understand. Did you weigh the same you weighed a year and a half ago? Like, was your blood pressure the same? I said, I don't understand. Why would you have done that? And they said, well, you didn't tell us not to. And I got, I thank God for Zoom, right? Because you can just sort of, be nice about it. But planning the study and sort of understanding what are microbes, what are they doing, and what do they naturally do? So some people get upset. They say, I tested in the spring, it was really high. And then I went back in December and it was low. I think your test doesn't work. And I was like, no, our test works. It's low in the winter. They're dormant. That's just natural. So we can talk about that more. Yeah.
It's interesting. I'm just trying to, how do I want to say it? Whenever I'm listening to anybody, in general, I'm always listening for someone that presses an aha moment in me that leads me to do something in the lab. So that's why this guy's kind of pressing you to see. I appreciate it. The other thing I'm noticing too is this is a compost tea. We've messed with this a little bit. We've seen huge multiplications of horrific biology. We used to send it in, and we always had trouble with the mail. And there's soil tests that supposedly you can send in, and they come back with biology numbers, and they have all sorts of things. Describe the validity of them. I mean, I've always been skeptical of them.
It's hard. So really, we have to say that the best thing you could ever do would be microscopy, okay? And that's getting a very expensive microscope and separating the microbes from the soil particles, which is our test. We have a way to do that, and looking at it under a microscope for a very, very long period of time and counting them one by one, okay? Now, no one's going to do that, okay? It's too hard, it's too expensive. And frankly, all of us have bad eyesight at this point, right? If you're a farmer, it's not like, so microscopy is really good. Our test mimics microscopy. A test like respiration measures carbon output of microbes, okay? Which is like measuring the number of people in a room by how much we breathe, okay? People, if they're running, breathe more. If they're, sleeping, they breathe less. So it's a proxy that has its place, and it's very good. If you mail soil in, you don't know what temperature the mail truck will be, and you don't know those things. And then a lot of the tests that rely on dried, reconstituted soil, right? We think the microbes that die aren't always the ones that come back, right? They can't prove that that happens. And while everything we know about the microbial community being interdependent means that you can't kill them all off and then just let the spores come back all at once in 20 minutes. That just doesn't make sense. That's what we preach, against. So there's no, we love the microbarometer. My mother developed it. She worked for 10 years alone in the basement. I'm glad I'm in agriculture because I can say I work for my mother's company and people don't think I'm a loser, because it's very common. And she did it because she said, look, lab tests for most people are out of reach financially. They have sort of logistical problems, right? And they're not as good as the lab people say they are. So she wanted to make one that any grower who was curious about their biology could do themselves, right? It wouldn't be quite as precise as the world's, as a microscopy. And if any of you want to learn microscopy, go ahead. But the DNA testing, that's the other thing that a lot of people talk about. We haven't genomed even a tenth of the microbes. So I think it has to get done and it probably will get done, but it's not like if I did a genome of you, I could tell you how you're going to die, right? It doesn't, it's not definitive information. So DNA testing is, $500 and doesn't at this point tell you enough about things, but that might change. So our test is designed to be done often, frequently, just to help you gauge what's going on with your soil in real time. And PLFA measures certain components of microbes, but it's hard to know if it measures all the microbes have them, that kind of thing. Does that answer it?
Yep, thank you.
Good. So I just want to say a little shout out. So the microbarometer, there's only 3 of you who can see it, but it's here. It's a little box. We manufacture it ourselves. My mother developed it. We make it in rural New York State. We do all the manufacturing ourselves. There's only 4 of us, so we ship it. And you can just order it direct. It's designed to be low cost. And any feedback we love to hear, even if you hate us. We still like to hear that. We don't love to hear it, but we want to.
So Laura, I'm David Little. I live in Utah. I've been
nice.
doing a lot of work in Georgia lately. And because it's starting to snow in Utah.
Yes.
So we'll have to go south. Anyway, I just wanted to make a comment. I've done a lot of both microscopy and sending samples to different microbiology laboratories across the US. This challenge of having your sample sit on a dock at a shipping station that's receiving direct sun at 110 degrees is real. And the sample will arrive at the microbiology laboratory absolutely dead, inert, no life. And you spend $100 to $500 per sample for a full microbiology assay on one sample that arrived dead. Not because there was no life when you pulled the sample, but because you cannot control shipping and what goes on between the time you drop it in the shipping station to when it arrives at the lab. So for that purpose, it's I think a definite advantage to be able to take a test kit to the field and establish a baseline based on your conditions, living soils. The other thing with the microscopy, it can be a little tricky to get the right dilution rates and to really spend the time to understand how to understand what you're looking at. And so I guess I just have one question. I'm new to the microbiometer. I'm actually just placing an order while you were talking.
Great.
But the thing that I wanted to ask, what are the right conditions, the right time of year to be creating our baseline and doing our our annual field checks to confirm whether what we're doing with cover crops and biostimulants and microbial amendments and compost extract and the five the 5 steps of healthy soil and regenerative soil practices. What's the right time of year to pull those samples with the microbiometer in order to be able to have an accurate tracking of the life in the soil?
So that's a really complicated question. And thank you for ordering. I want to make sure I have a bunch of coupon codes too. So did you hit send yet? So I hope not. If you use coupon code BERG15, you can get a discount on it. Or if you sign up for our newsletter, I think you can get 20% off. So save some money. It's free shipping. Yeah, it's a baseline test. So you're not searching for a particular number. And the weird answer is the microbes will naturally cycle through the year, right? And each year is different. It's dry, it's wet, it was a late spring, it was a hot fall. So really what you want to do, and I'm hoping to get, we got these great new app developers. We're hoping to be able to have people sort of chart it. So it's almost like I'm an accountant by trade. So when you look at financials, you have expectations, right? You say, this is what I think should happen, right? Based on what I know intellectually, right? And then I go out and I say, is it different than what I expected? And so my ideal would be that all growers sort of know, they take enough tests at different times of the year and note what's happening, right? Because every December 15th is not the exact same condition, right? But if you note cold, snow, bare ground or something, you have to get a feel for it, right? So you should test sort of in general, you test sort of before you do anything, right? And then you want to test right before planting, right? I think when the plant really starts to photosynthesize, you want to see what's happening. And then as you do treatments, you want to see, you don't want to do it right after you do a treatment. You want to do it a bit afterwards. And then you want to see how long that treatment, like Matt was talking about, the treatment that he did to that soil and then treated badly by putting in a cabinet still benefited the soil in the long run. So you want to say, am I getting 2 months later it better than it would have been? And then you do want to say, okay, when the plant goes to flower, what kind of dip do I get? And then what happens when things stop growing? So you do want to periodically do it. We're trying to get the test much cheaper, so it's only a couple bucks so each time, so people can do it a lot all over the place. Because again, the more data points you have, the more you can sort of get an average and a trend. Because there'll always be something weird happening,
So in the springtime, is there a specific soil temperature that I can use as a guide? As we're coming out of our last frost, the ground's thawing out, and I'm getting ready to plant. Once that soil temperature hits, 65 degrees or 60 degrees or 50 degrees, is there a soil temperature that says, okay, the microbes should be engaging or active enough to get a reading?
I don't know if someone can opine that's good. I think you'd have to test a bit at different temperatures to see when does it start, right? And it might depend on both the amount of daylight, the moisture, and if anything's growing, right? If things are growing, you'll always have some microbial activity, right? No matter what. And if you have roots decaying in the soil, you'll have some activity as well. Again, which is why people are so crazy about cover crops. I wish I knew more. I'm not an agronomist. We have a soil scientist who we're training or having her get trained on this. And we're hoping to get more of that information to be able to help growers. But it feels a little bad right now that we don't know everything. But it's kind of an exciting time, I think, in agriculture, because really the farmers and the growers are the ones that are driving a lot of the change. And the knowledge, right, is coming up. We've seen what the academics do. It's not that helpful.
Thank you.
Thanks.
All right. Thanks a lot, Laura.
Yeah,
That
thank you for
was- that
having
was
me. I really,
awesome.
and my email is very boringly laura@microbiometer.com. But on our website, if you forget it, it just says message us. And all four of us in the company get all the emails. So it's just info@microbiometer if you forget it and you want to ask me anything.
That's how I first contacted you.
Yeah, we are, but it's Laura@microbiometer, and our website is microbiometer. And I think that the coupon code BERG15 works as well too.
Awesome.
Thank you. I
Yeah,
appreciate it.
that was great. That was very informative. Thank you. So thanks everybody for listening in. Our next conference call will be on December 8th at 9 p.m. Eastern Time. Thank you for tuning in, and I hope to see you or hear from you in a month. Take care.
Thank you, Laura.